Okay, we're only going to touch on a couple of things from chapter 23 on enzymes. We're going to look at enzyme catalysts, enzyme terminology and activity, which is pretty important, various examples of enzyme regulation, how enzymes are used in medicine, and I'm just going to mention transition state analogs. The only purpose of me showing you this picture is because I want you to understand how complex enzymes are. This is the enzyme that your body uses, cytochrome C oxidase, that is activated during respiration when we take in oxygen. And you can see that it's pretty complex. Remember, enzymes are proteins, and proteins can be made up of alpha helixes, beta pleated sheets, random coils. Pretty impressive, actually. Enzymes are considered biological catalysts, meaning they speed up a reaction, with the exception of some RNAs. RNAs have their own ribozymes that catalyze their own self-cleavage, meaning that they're special ones that break the RNA in certain places. Enzymes can increase the rate of a reaction by a factor of 10 to the 9 to 10 to the 20 over an uncatalyzed reaction. So that's pretty fast. Some catalyze the reaction of only one compound, meaning that they are very specific, while others can just are more broad spectrum. Here you can see that urea is catalyzed by urease to form ammonia and carbon dioxide. The majority of enzymes are globular proteins. We went over this in the last unit, which means that they're soluble in water, and they're mostly used for non-structural purposes. So basically, most of the enzymes that we're going to talk about are considered globular proteins. They're not structural proteins. Enzymes do not change the position of equilibrium. That's really important. The only thing is they just speed it up. That's it. They're not going to make it go more in the forward direction or more in the reverse direction. The reaction would still take place without them. They would just occur very, very slowly. Enzyme catalysis. All this refers to is the acceleration of the reaction by the catalyst. So here you're seeing trypsin catalyzing the hydrolysis of a peptide bond formed by the carboxyl group of lysine and arginine. So lysine, okay, and arginine has this group where you see the blue arrows. Let me get my pen. Remember, that's your peptide bond. We talked about that in the last unit. This was a COOH bond and an NH2 bond. Hydrolysis, meaning we removed the water, and we were left with the COO bond and the NH bond. So we started with a carboxyl group. Now we have a carbonyl group left. Trypsin catalyzes that reaction, making it happen much more quickly. And that's all that this is showing you. And they're showing you that it happens in more than one place when the amino acids are linked together. Here I'm going to give you an example of enzyme specificity. And one of those is with muscle relaxants. A lot of people have to take muscle relaxants. So, nerve transmits signals to muscles. Acetylcholine, which is a neurotransmitter, binds to a receptor in the muscle end plate, causing it to contract. Acetylcholinesterase causes the hydrolysis of the acetylcholine, removing it from the muscle end plate, and prepares the muscle for the next signal. Muscle relaxers contain succinylcholine, which binds to the receptor site normally occupied by the acetylcholine. The acetylcholinesterase can hydrolysize the succinylcholine while attached to the receptor site, but only very slowly, so the muscle stays relaxed. So let me break that down a little bit easier. So enzymes can work in a variety of ways. In this case, the catalyst is the acetylcholinesterase. It removes water from the normal. It adds water to the original acetylcholine that is there, causing it to change and dislodge from that end plate. This now opens up the spot for the succinylcholine found in the muscle relaxer to bind to that receptor site. So what happens is that the impulses that send message to the brain to allow the muscle to contract are going to happen at a very, very reduced rate, because the succinylcholine is not affected in the same manner as the acetylcholine by the acetylcholinesterase. Enzymes are broken down into various categories, and they're usually categorized by either the reaction or reactions, if it's more than one that they catalyze. So, for example, lactate dehydrogenase is responsible for the conversion of pyruvate to lactic acid in the glycolysis process. And acid phosphatase actually functions to liberate phosphate under acidic conditions. It's made in many parts of our body, but it's most prevalently known in males in the prostate gland. And actually, when males have cancer that spreads to other parts of the body, this acid phosphatase increases in quantity in exponential amounts. You don't have to know what they do. I'm just giving you some specific examples of enzymes that are not broad spectrum. So enzymes are classified into 6 major groups. We have oxidoreductases, which work in oxidation reduction reactions. We have transferases, so group transfer reactions. Hydrolases, which those are probably the ones we've talked about the most, hydrolysis reactions. Lyases, okay, the addition of 2 groups to a CC double bond, or the removal of 2 groups to create a CC double bond. So we looked and we talked about organic chemistry. When you have a double bond between 2 carbons, you reduce the number of hydrogens or other groups that are attached. The other way that can work is you can break a double bond, make it a single bond, and then add more groups. Because the bottom line is a carbon can only form 4 bonds. So that's the job of the lyases. Isomerases, they're involved in isomerization reactions. And all that is a fancy word for making isomers. So you have one structure and you want to create a different structure by rearranging it. This enzyme can make it happen faster. Ligases are responsible for the joining of 2 molecules. I just gave you an example of that with lysine and arginine on a previous slide. In the first example here, you have oxidation reduction going on by the lactate dehydrogenase. you have the NADH being oxidized to NAD plus and the hydrogen being reduced. So the function here is to take the pyruvate and turn it into lactate by adding two H's, one from the NADH and the other from an acid. And the anion of the acid is just a spectator, so it's not listed here. So you added an H to the C bond, and you added an OH, an H to the O bond. So you basically took the carbonyl group and turned it into an alcohol. In transferase, you're taking one functional group off of something and putting it on something else. You're exchanging them. So on the left, you have aspartate and you have alpha ketoglutarate. And using aspartate aminotransferase. So amino refers to the group that it's transferring. So if you look at the aspartate, we took the NH3 off of that and we put it onto the alpha ketoglutarate where the carbonyl group is. And what that resulted in is changing the structure, and because you exchanged that functional group, you now created oxaloacetate and glutamate. So transferase just simply exchanges one functional group for another. So I talked about this a little bit on the previous slide. Hydrolases, here you're taking the acetylcholine that's found at the receptor site, and in the presence of acetylcholine esterase, you are breaking that down into acetate and choline, so it dislodges from the receptor site, which will now allow succinylcholine to take its place and allow the muscle to relax, because it cannot be affected by the acetylcholine esterase in the same manner. So remember, liases, I said, can either add a double bond between carbons or remove a double bond between carbons. In this case, you have cis-actinate, and it's turned into isocitrate by the removal of the double bond between two of the carbons and the addition of water. So when you do that, you take the double bond out between the two carbons and you add water, so you're technically turning it into an alcohol. In isomerase, remember, you're taking the original compound, so in this case glucose 6-phosphate, and you're turning it into fructose 6-phosphate. And you do that by rearranging the functional groups. Both of these have the same amount of carbons, hydrogens, and oxygens, and phosphate groups. I don't expect you to know any of these in particular. I just want you to understand what's happening. And then the last one here, they're just giving you an example of ligase. You're taking different fragments and joining them together. So ATP, L-tyrosine, and tRNA. And then you're making the chain bigger, so to speak. So a lot of times you'll see ligase reactions include the word synthetase. And synthesize means to make. So you're basically making new molecules. Okay, enzyme terminology. These are important, people. And we're going to be talking about the structure and how the enzyme reacts All right, apoenzyme is the protein part of an enzyme. You can remember it because it has the P in it. Cofactor is the non-protein portion of an enzyme, and that part is actually necessary for the catalytic function. Okay, it helps it go faster. A coenzyme is a non-protein organic molecule. This is often a B vitamin that acts as a cofactor, so it helps turn the substrates into products. So it's similar, okay, to a cofactor, as it's also non-protein, and it acts as a cofactor, but it's not actually part of the enzyme. It's in addition to. Substrate is the compound or compound whose reaction an enzyme catalyzes. So the substrate is what the enzyme is working on. And the active site is the portion of the enzyme to which the substrate binds during the reaction. So the active site is located on the enzyme. The substrate is on the compound that the enzyme is working on. Okay, so here we have our enzyme and substrate complex. You can see here your apoenzyme, so that is the protein part of the enzyme. Here you have your substrate molecule. This is what the enzyme is going to act on. You have a coenzyme, right? Coenzyme is non-protein. It's going to help it go faster. And the reason why the coenzyme is here is because it fits into the active site. And then your coenzyme and substrate fit into that active site, forming the enzyme substrate complex. It does its thing and then releases the new product or keeps going in the reaction depending on the function of the enzyme. Okay, some important terms in enzyme chemistry. Activation, any process that initiates or increases the activity of an enzyme. Inhibition, any process that makes an enzyme less active or inactive. So I don't know if it's classified really, the succinylcholine that's found in muscle relaxants as a true inhibitor, but it acts in an inhibitor way because it binds to the end of the receptor site, and the acetylcholinesterase cannot affect it in the same manner as it does the acetylcholine, which thereby relaxes your muscle. These two next words are extremely important: competitive inhibitor and non-competitive inhibitor. A competitive inhibitor is a substance that binds to the active site of an enzyme and thereby preventing the binding of the substrate. Non-competitive inhibitor is a substance that binds to a portion of the enzyme other than the active site, so the apoenzyme portion, and thereby inhibits the activity of the enzyme. As you can see, there's something at the end of this called enzyme review. I cannot embed that in this video because it's copyrighted material, but it's imperative. It's a really short 5-minute video that you go back to the original PowerPoint in Brightspace and click that enzyme review link. It really goes through the difference between inhibition, competitive inhibitor, and non-competitive inhibitor. So please take the time to go back and watch that. Enzyme activity is a measure of how much a reaction rate is increased. There are 3 ways that we can look at this. Enzyme catalyzed reactions are affected by enzyme concentration, substrate concentration, temperature, and pH. So what this is showing you is basically the effect of changing the enzyme concentration on the rate of a reaction. If substrate concentration, temperature, and pH are all constant, and the only thing that we're changing is enzyme concentration, you can see that if we increase the enzyme concentration, we increase the rate of the reaction linearly. That's all this is showing. Okay, so the opposite here. Now we're going to keep enzyme concentration, temperature, and pH constant, and we're only going to change substrate concentration. I see a typo and I'm laughing over here because I can't change it because it's embedded in the video, but if we increase the substrate, keeping everything constant, the curve doesn't change, the curve changes, and it's not linearly. It increases continuously, but then it's going to level off, and eventually this would become a straight line because basically all of the substrate is bound. You don't have any more substrate left. This is pretty interesting, and you should have learned this in bio if you have taken bio, but if not, I do need to go over it. So enzymes are temperature specific. So there is a range of optimal working function for enzymes. So, and if you go outside that range, there's two things that can happen. Either one, the enzyme functions at a slower rate, or not at all, because you may have denatured the enzyme and it cannot be repaired. And that's because when you change the temperature too drastically, you're actually going to change the shape of the enzyme. And lastly, pH affects the enzyme in a similar manner. There's an optimal pH range for the enzyme substrate to work. And if you're outside that optimal range, it changes the shape of the enzyme. Again, either it's going to make it work very slowly, or it could actually denature it and it not work at all. So we already looked at this earlier, and I kind of talked about this, so I'm just going to review it. The enzyme substrate model is referred to as lock and key because the enzyme and substrate are specific for one another. You can't just take any old substrate and put it in an active site. It actually has to be specific for the enzyme. And the two of them combined in the last picture are called the enzyme substrate complex. Now there are broad spectrum enzymes, I like to refer to them as, and these usually function in an induced fit model. And you probably did not talk about this in bio if you took it. The active site actually becomes modified, okay? It's flexible to accommodate the substrate. And this is the induced fit model, not lock and key. Lock and key is specific. This is a review of the competitive inhibition. Hopefully you watched the video, or you will watch the video. In competitive inhibition, you have an inhibitor that binds to the active site in the enzyme, and therefore the substrate cannot enter because the shape has been changed. In non-competitive inhibition, the inhibitor binds to the apoenzyme, the protein portion of the enzyme, and causes the shape of the active site to change because it's bound to the apoenzyme portion, and therefore the substrate cannot enter because the shape of the active site is changed. The other possibility is that it enters the active site. As you can see in the bottom portion, you don't have a good fit, so catalysis cannot occur. In a later part in this lesson, I'll talk about allosteric inhibition, which actually changes the active site enough where the substrate can't enter at all, but that's a slightly different type. This probably isn't as important as some of the other stuff I've mentioned, but I'm just showing you the difference between how the rate of reaction changes based on whether you have a competitive inhibitor, a non-competitive inhibitor, or no inhibitor. In looking at the purple and red line, which is your inhibitor versus competitive inhibitor, you can see that the maximum rate is still achieved. However, the competitive inhibitor does so, it's achieved at a low concentration of substrate as versus no inhibitor. And it's achieved, that rate is achieved, the no inhibitor is achieved at a high concentration of substrate. Non-competitive inhibition does not achieve the same rate of reaction. Again, this is just showing you the difference in rate. I don't think it's anything that you need to worry about. So just kind of a quick review. Both the lock and key model, specific enzyme, and induced fit model, broad spectrum enzyme, emphasize the shape of the active site. In the induced fit model, however, the chemistry of the active site is most important. 5 amino acids participate in active site in more than 65% of the enzymes studied to date. So those 5 are histamine, cytosine, aspartame, arginine, and glutamine. 4 of these amino acids have either acidic or basic side chains. And the 5th has what's called an SH group or sulfhydryl group. Again, I don't think that so much is important in the overall scheme of things. So what does an enzyme actually do? How does it affect the rate? So here you have a potential energy diagram. And what happens is you have reactants and you have products, okay? So here are your reactants. Here are your products. This is called transition state, or what's called the activated complex. The activated complex is an intermediate state. It's where the reactants and products have come together, but the products are not fully formed yet. So think of riding a bicycle up a hill. When you ride a bicycle up a big hill, it takes an awful lot of energy. So that's your activation energy. Activation energy is the amount of energy needed for the reaction to start. Okay? Here's your reactants, here's your products. Here's the amount of energy needed to get from the reactants to the activated complex in an uncatalyzed reaction. It's the same thing that you saw in the diagram on the left. Now, when I add a catalyst, right, reaction goes faster. Why? It creates an alternate pathway for the reaction to occur, still starting at the reactant line, finishing at the product line. But now it takes less energy. So it's like riding your bike up a small hill. So the way that the enzyme works is by reducing the amount of energy that's needed to start the reaction so that it can take place. Enzymes are often regulated by environmental conditions. An enzyme regulation process where the products of a series of enzyme catalyzed reactions inhibits an earlier reaction in the sequence. This is called feedback inhibition. So what happens is you have a reaction that takes place, right? Enzyme 1 produces product B. Enzyme 2 does its thing, produces enzyme C. Enzyme 3 does its thing, produces product D. At this point, the production of D goes back and inhibits enzyme 1, saying, ding, 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 we have enough product right now. Don't continue. This inhibition may be either competitive or non-competitive. And an example of this is ATP in cellular respiration. I cannot show you this now because it's a link. So if you want to see this, go back to the original PowerPoint in Brightspace and click that link. So enzymes are like really interesting creatures, if you would. They're just so capable of so many things, and I can't imagine like how our bodies would even function without them. They're just incredible. And one thing about enzymes is they're not used up, okay? You can use an enzyme over and over and over again. So an interesting one is something called a proenzyme. In this case, a zymogen. It's an inactive form of the enzyme. And in order for it to become activated, it has to have part of its chain hydrolyzed and removed. So an example of this is trypsin. It's a digestive enzyme. So it's synthesized and stored in our bodies as trypsinogen. And it has no activity when it's in that form. It only becomes active after a 6 amino acid fragment is hydrolyzed and removed from the N terminal of its chain. So remember how our chain's formed, N terminus to C terminus. So it's having the front part of the chain removed. Now the removal of this small fragment changes not only the primary structure, but also the tertiary structure, allowing the molecule to achieve its active form. This is important. Why? Because it only becomes active once it enters the digestive tract. So basically, when you eat something, right, the body's going to signal to activate the enzyme. When you're not, then there's no need for it to be working, so to speak, in layman's terms. Okay. Allosterism. Enzyme regulation based on an event occurring at a place other than the active site, but that creates a change in the active site. And I do have a video on allosteric regulation. And I actually, when I did this in class, I actually just showed them in class. What I think I'm going to do, since there's 3 links in this video, I'm just going to put them in Edpuzzle right after this video. So watch the chapter video first, which obviously you're doing, and then I'll put them so that you can see them also as reinforcement if you want to. This way you don't have to go back to Brightspace and find them. An enzyme regulated by this mechanism is called an allosteric enzyme. So allosteric enzymes often have multiple polypeptide chains. There's 3 different types. You have negative modulation, so that's the inhibition of an allosteric enzyme. You have positive modulation, which is the stimulation of an allosteric enzyme. And then you have a regulator, a substance that binds to an allosteric enzyme. So those are the things that are needed to be involved in allosteric regulation. So the next slide or so takes a look at how that happens. So here we have the binding of a regulator to a site other than the active site, and it changes the shape of the active site. So you might be thinking right now that this is a lot like non-competitive inhibition. How is it different? Because they're both binding to the apoenzyme, changing the shape of the active site. So let me say this. All non-competitive inhibition, which is what I talked to you earlier, is a form of allosteric inhibition. But not all allosteric inhibition is non-competitive, because in some instances the allosteric inhibition can be so severe that it actually prevents the substrate from binding to the active site at all. Whereas in non-competitive inhibition, the substrate can still bind to the active site, but the type is not fit, so therefore the reaction can't take place. I hope you understood that. I said it as basic as I could. There's also something called protein modification, and I'm just going to breeze through this. I'm not holding you responsible for it. It's the process of affecting enzyme activity by covalently modifying it. This happens in phosphorylation and dephosphorylation of the pyruvate kinase. The pyruvate kinase is the active form of the enzyme. It is inactivated by phosphorylation to produce pyruvate kinase phosphate. And this is all happening in your changing of ATP to ADP in glycolysis. Isoenzyme, iso means same, so they're also referred to as isozymes, and this is an enzyme that occurs in multiple forms in different tissues. So think of it as an isomer, right? Iso, same number of hydrogens, carbons, whatever's involved, different shape, all right, depending on the tissue that it's in, but they all catalyze the same reaction. So lactate dehydrogenase, LDH, catalyzes the oxidation of lactate to pyruvate in glycolysis. The enzyme is a tetramer of H and M chains. The H4 is present predominantly in heart muscle, while the M4 is present predominantly in the liver and skeletal muscle. They both do the same thing. Okay, and I'm not getting like in depth on this. I just wanted you to understand how the isoenzymes or isozymes work. They work to catalyze the same reaction, but they have slightly different structures depending on what part of the body they're located in. And I have an animation of LDH here too, which I will post as a separate link. So this is just showing you in more depth the isozymes of lactate dehydrogenase depending on where they are and what tissue they're found in. So you can see in the liver and in the muscle you have the M4 form, where in the brain and red blood cells and kidney you have the MH3, but in the kidney you also have the MH4. That's also this. It's all this is showing you. All right, I'm not holding you responsible for this. I want you to realize that there are different forms of the same enzyme that catalyze the same reaction depending on what tissue they're located in. Abnormally high or low levels of enzymes in various body fluids signals either the onset of certain diseases or their progression. And I mentioned this earlier in the video with the prostate gland and cancer. So here are some of those different enzymes. So if you have a high level of alanine aminotransferase in your blood, it could be the signal of that you have hepatitis. Acid phosphatase, I already spoke about, prostate cancer. Going down, if you have normally high levels of lactate dehydrogenase, LDH, creatine phosphokinase, CPK, or phosphohexose isomerase, PHI. This could signal that you've had a heart attack. So when someone goes into the hospital, they're running blood chemistry on all of these different enzymes to see what's going on, to give them an indication of what might have happened, or an indicator of what is happening. Transition state analog. What this is, I'm just going to briefly cover it, a molecule whose shape mimics the transition state of a substrate. And the best way to see it is to look at the small video clip that I was going to show you on molecular mimicry. And lastly, an Ezyme is an antibody that has a catalytic activity because it was created using a transition state analog as an immunogen. And also a video clip on using Ezymes in the HIV vaccination. So I know I flew through this. Again, I'm going to post all of the links in this PowerPoint that you would have seen in class as separate links in Edpuzzle so that you can view them if you want to. Please remember, our exam is Wednesday, 3 to 5:30 in our normal room. Please bring any work that you owe me and labs, etc. Okay, thanks very much. Oh, and if you had something that you needed to give, I guess, Dr. Lewis, and you can't find him or see him, please bring it to me.